Welcome to video 7, and in this video we are looking at the origins of cells, unit 1.5 in topic 1 of cell biology, IB standard level bio. Okay, before we start, we need to think back to some of our previous units, and we need to think about this whole concept of the cell theory. So the cell theory stated these 3 points. All organisms are composed of one or more cells. Cells are the smallest units of life, and all cells come from other pre-existing cells. So this is our current theory on cell biology. Now, we need to really understand this to be able to build further on this in this unit. What other things do we already know? Well, part of that cell theory was that all of the cells arise from pre-existing cells. And for that to happen, all cells of an organism therefore must contain the same DNA. So some process must occur for these cells to divide and contain identical DNA, and we'll look at that in the next unit. Multicellular organisms, so they're those organisms with more than one cell, develop from what we call gametes. Okay, so that's just some pre-existing information that we should have that we can now build on in this unit. So, with all that in mind, let's think of this question. Where did the first cell come from? Now, there is this continuity of life on Earth, okay? So from its origins to the cells in our bodies now, okay? So in organisms on Earth today, there are these cycles of nutrients throughout Earth and its history. So we need to bear that in mind, okay, as we move through this unit. However, the early scientists obviously didn't know this, so they had their own ideas and theories on how the first cells developed. Okay, and the theory of spontaneous generation was the leading theory for many scientists for centuries. Okay, and that theory stated that the formation of living organisms came from non-living matter. So the cartoon here is showing no cat, and then the cat spontaneously appears from beams of sunlight. Okay, so that's the sunlight spontaneously generated cats. Cartoon, but that was the idea, that organisms spontaneously generated from non-living matter. Now, this was believed to be the way until Louis Pasteur completed his famous experiments disproving the existence of spontaneous generation. So let's have a look at that. Okay, so Louis Pasteur, born in 1822, died in 1895. And in the 19th century there, Louis, a French biologist, designed an experiment to show that bacteria could not spontaneously appear in a sterile broth. Let's have a look at his method. Okay, so first of all, he boiled a nutrient broth to sterilize it. Then he placed this sterile broth into 3 flasks. So if you see the image there, we've got an open flask, we've got a sealed flask, and then we've got a flask with the U-bend full of water in it. Can you predict what the results were? So this broth was incubated over a period of time in these flasks, and a sample of each flask was then transferred to a plate containing a solid medium and was further incubated. The only flask that showed the presence of bacteria was the open one. The other 2 showed no evidence of any bacterial growth at all. So the sealed one and the one with the water in the U-bend showed no evidence of bacterial growth. This indicated to Pasteur that the concept of spontaneous generation was wrong. Why is that? Well, let's think. If there was bacteria in all three, well, how did it get in there when the broth was sterile from with the sealed beaker and the one with the water and the U-bend? How did it actually get in there? Alrighty, so this led to the new question, or the question, where did the first cell come from? Because Louis Pasteur has now proven the falsification of the spontaneous generation theory. So where did the first cell come from then? The origin of the first cells. One opinion, they arrived on Earth from elsewhere. The more widely accepted opinion, they must have arisen somehow from some non-living material. Unlikely that a complex cell sprung up out of non-living material. So had some non-living material, and next thing we've got a complex cell. So it's unlikely that that has happened. It's theorized that the first cell was produced over a number of stages, okay, and over many millions of years. Let's have a look at these proposed stages.
Scientists in Georgia think they might have found out how all life started on Earth. Yeah, we're about to get real. Strap in. Anthony here for D News, and if there's one question we all want the answer to, it's where did we come from? And now researchers at the Georgia Institute of Technology think that they have figured out a crucial component of the answer. So, about 3.5 billion years ago, the Earth is a lifeless, oxygen-free environment, and then at some point, bacteria form and start converting all the carbon dioxide, methane, and ammonia, and turning it into oxygen. But we've never been sure where that bacteria came from. Earth's oceans at the time were filled with all kinds of minerals, including a lot of iron. If you've heard the term primordial soup, that's what we're talking about. Earth's original atmosphere and mineral-filled ocean. So the idea is that in the soup are all the things that you need to make RNA. And then the RNA interacted with something, some sort of chemical reaction or spark, and started creating all of that bacteria and all life on Earth. Now, there have always been a couple problems with this theory. First of all, RNA isn't really capable of making life on its own. Your DNA has all the instructions for making you and the ability to transfer that information. RNA basically acts as a messenger. It takes the instructions from your DNA and it uses them to make proteins. Without DNA and protein-based enzymes, RNA doesn't survive. So the second thing is that there was nothing in the environment that anyone could find that would jumpstart the ability to create life. I mean, there are theories that lightning did it. Another theory is that there was an impact from space, but that's mathematically silly. So here's what the team in Georgia found. If they recreated the primordial soup in a chamber, RNA actually changed. It started behaving differently in an environment without oxygen. It was almost like it reactivated a function that it hadn't used in billions of years. It shape-shifted and all of a sudden became an instruction carrier, kind of like DNA. And when they teamed it up with that iron from the soup, it performed a single electron transfer. That means it jump-started the reactions needed for photosynthesis and respiration. RNA, together with iron, created the basis for an eating, breathing thing. Boom, life on Earth. Bacteria forms, oxygen into the atmosphere. 3.5 billion years later, we're all reblogging sloth gifs on Tumblr. Now, there are a few holes in this theory. If you asked, hey, where did that RNA come from in the beginning of the world? You are asking the right question. See, all the components of RNA were in the soup, but they're not components that like to bond easily, though there have been some successes in making it happen in a lab. But how crazy is that, right? RNA, this molecule that we thought was so simple, we thought we knew everything about, way more adaptive than we thought. So maybe there's a way to create it that we haven't thought of, and then we'll be even closer to figuring out where we came from. The origin of all life. That is pretty rad. What is the biggest question you want science to answer? Let me know down below, and subscribe for more D News.
So, moving on from that, steps involved in the production of the first cells. What is the theory? Summarizing what we've just seen. Production of carbon compounds such as sugars and amino acids. We had the assembly of the carbon compounds into polymers and formation of membranes, okay, followed by the development of a mechanism for inheritance. Alrighty, let's have a look at each of these steps. So the production of carbon compounds such as sugars and amino acids, and we're going to be having a look at this Miller-Urey experiment, okay? So Stanley Miller and Harold Urey created an experiment in which they passed steam through a mixture of methane, hydrogen, and ammonia. This was designed to simulate the early atmosphere of Earth hundreds of millions of years ago. And electrical discharges were used to simulate the lightning that would have been occurring in that atmosphere. They found that amino acids and other carbon compounds that are required for life were produced through this experiment. Okay, so the next step would be the assembly of carbon compounds into those polymers, so those long chains. So the predicted site for the formation of carbon compounds is deep in sea vents. Okay, these vents gush hot water carrying inorganic chemicals like iron sulfide. These chemicals are a readily accessible source of energy for the assembly of the carbon compounds into polymers. For example, our phospholipids that we've looked at in previous units. So our third step then, we've got our phospholipids, is the formation of our phospholipid membranes. And we know that they formed into our bilayers. So this would have allowed different internal chemistry from that of the surrounding areas to develop. And then our fourth step, the development of a mechanism for inheritance. Okay, DNA is the mode of inheritance in living things, we know that, and enzymes are required for DNA to be able to replicate, so that we will look further into DNA replication, but we already know that enzymes are required to do this, but DNA is required to produce those enzymes. So which came first, the DNA or the enzymes? It's a whole chicken and the egg question. But thinking back to that video, RNA may have been the earlier mode of inheritance, as it can store genetic information, it's self-replicating, and it can act as an enzyme or catalyst itself. So we heard in that video about how the mRNA was formed. Okay, so how did complex eukaryotic cells form then? We've looked at the basic formation, or the proposed basic formation of cells, but there we're looking at very basic cells there. How did our complex eukaryotic cells form? So the current theory is endosymbiosis, or the endosymbiotic theory. Okay, let's look at that a little bit further. So in the endosymbiotic theory, it states that mitochondria were once free-living prokaryotic organisms that had developed the process of aerobic respiration. Larger prokaryotes that could not perform aerobic respiration then engulfed the mitochondria through endocytosis. And we looked at endocytosis in a previous video. Now, instead of killing and digesting the mitochondria, they allowed them to continue to live within their cytoplasm. As long as the mitochondria could grow and divide as fast as the host cell, it could live inside the larger cell indefinitely. This relationship is called a symbiotic relationship, as both organisms are living together. And as they both gain benefit from this arrangement, we call it mutualism. So the mitochondria can provide energy for the larger cell, and the larger cell provides the energy for the food for the mitochondria. So thinking of that glucose required for the respiration to occur. This mutualistic symbiotic relationship allowed organisms to survive beyond those prokaryotic types which had not undertaken the process. This is also true for the chloroplast. Okay, so thinking of both the mitochondria and the chloroplast. They both have their own genes on a circular DNA molecule like that of our prokaryotes. They have their own 70S ribosomes, a size typical of that found in prokaryotic cells. They can transcribe their DNA and use the mRNA to synthesize some of their own proteins. And they can only be produced by division of pre-existing mitochondria and chloroplasts. Okay, where are we up to? So we've crossed quite a few bits off there. I hope you've enjoyed this video, and I'll see you soon in cell division.